The overall goal of this procedure is to observe the effects of the depletion of specific genes of interest on cell migration.
This method can help us answer key questions in cancer metastasis, such as cancer cell migration.
The main advantage of this technique is that it allows the dynamic cell migration to be monitored in real time.
We first had the idea for this method when we were observing cell morphology changes upon the depletion of TIP60, a tumor suppressor.
Begin by seeding 1 million breast epithelial cells in 3 milliliters of culture medium per 10 centimeter dish. Add 20 minutes room temperature incubated siRNA mixture drop by drop to the experimental cell culture. And manually shake both dishes to ensure that the cells are evenly distributed before placing them into a 37 degrees Celsius incubator. After 6 hours, replace the supernatants with 8 milliliters of fresh culture medium per dish. And return the cultures to the incubator. The next morning, replace the supernatants with 3 milliliters of fresh culture medium per dish. And add a second batch of siRNA mixture to the experimental cell culture as just demonstrated. Shaking the dish to ensure that the reagents are fully mixed before returning the cells to the incubator. Then replace the supernatants with 8 milliliters of fresh culture medium after 6 hours